students, and today we are going to discuss the questions we can expect from the topic the photosynthesis in higher plants. Okay, see the first question. So NADPH is generated through, and see the choices: photosystem 2, anaerobic respiration, glycolysis, and photosystem 1. Okay, so the question is about the NADPH formation in the photosynthetic process. As we have seen in the introduction of the photosynthesis, there are two major systems involved in the system of the light reaction system, photosystem 1 and photosystem 2. Okay, so they have their own the pigments, and they are going to accept the light energy in the form of photons, and they are going to excite, and the excited energy they are going to activate the molecules. Okay, and they are going to produce the release of electrons, and those release of electrons or released electrons entered into the cycle of molecules or complexes, and they are going to produce the energy rich carriers of molecules like ATP and NADPH. Okay, so this is the common thing you have to remember about the photosynthesis. There are two major reactions as we have discussed: light reaction and dark reaction. In case of light reaction, they are going to produce the energy rich molecules of ATP and NADPH. In case of dark reaction, and they are going to use the particular molecules ATP and NADPH will be used in the dark reaction to produce the carbohydrate in the dark condition by using the enzymes. Okay, so this is what you have to remember, and the question over here is not talking about the ATP and NADPH, it is talking about only the NADPH. Okay, so in the light reaction we have seen two different kind of reactions. Okay, so whenever you are going to produce the high energy molecule, we are calling that reaction as the energy producing reactions, and over here we have seen two different cycles: cyclic phosphorylation and non-cyclic phosphorylation. Okay, so over here, what do you mean by non-cyclic phosphorylation? Over here the electrons will not be backed, or will not be in a cycle to the particular reaction, and it will be delivered to the production of NADPH. Okay, so in case of cyclic phosphorylation, there will be no production of the particular NADPH, because over here there will be no involvement of enzyme called NADP dehydrogenase. Okay, so in the absence of the particular enzyme and in the absence of some other system, over here cyclic phosphorylation going to produce only the ATP molecule and not NADPH molecule. Okay, and this is the second point you have to remember, and the question over here it is talking about NADPH generated through. Okay, so over here you have some keywords: photosystem 1, photosystem 2, and anaerobic respiration and glycolysis. Okay, and you can easily eliminate the glycolysis and anaerobic respiration. You have to choose from only the PS2 or PS1. As I told you, there are two major reactions over here: cyclic and non-cyclic phosphorylation. So what do you mean by non-cyclic phosphorylation? Over here the involvement of PS2 and PS1. This is what you have to remember in case of non-cyclic phosphorylation. So what happens over here, as I told you, starts from the light, the photons will be absorbed by the pigments of the photosystem 2. Okay, and it is going to excite, electrons going to release from here. Okay, and the electrons will be accepted by the electron acceptor, and then it will be transferred to the photosystem 1. Over here also there is a reaction center, it is going to absorb the particular electrons released by the particular PS2, and in between the PS1 and PS2 there is a electron transport chain, and that is going to produce the ATP molecules. That is what you have to remember over here. Okay, and the point you have to remember over here, in the PS1 again it is going to accept by the electron acceptor over here, and these electrons, okay, the electrons accepted by the particular PS1 system will be delivered to, or it will be transferred to the final acceptor molecule NADP plus over here, and it is going to produce NADPH molecule over here. Okay, so the point you have to remember over here, the answer for you over here, the PS1. Why? As I told you, here only it is going to produce the particular NADPH molecule. Suppose you have a choice over here like PS1, PS2 and for example instead of glycolysis you have non-cyclic phosphorylation. You have to choose non-cyclic phosphorylation because the NADPH is the product of non-cyclic phosphorylation. In case of cyclic phosphorylation, there will be no production of NADPH, only the production of ATP. This is what the major differences you have to remember in between the particular production of ATP and NADPH. Okay. And now see the next question. Number of Calvin cycles required to generate one molecule of hexose. Okay. So it is talking about a production of one sugar molecule. Over here hexose. Hexose means 6 carbon sugar. For example, glucose is a hexose. Okay. So production of one molecule of hexose requires how many number of Calvin cycle? Okay. And we have seen Calvin cycle. Calvin cycle is nothing but C3 cycle. It will happen one in the C3 plants. Okay. So what will happen over here? What do you mean by Calvin cycle? It is a kind of the carbon dioxide fixation. Okay. Over here the carbon dioxide from the atmosphere, it is going to be fixed as a sugar. Okay. So over here there it's a continuous cycle of oxidation and reduction reactions. Oxidation and reduction reactions. Okay. So what happens over here? Oxidation and reduction, as I told you. So there are two major molecules involved over here, as I told you, carbon dioxide and water. Okay. So here you have to remember one thing, the water is being oxidized and the carbon dioxide is being reduced. This is what you have to remember. Water is being oxidized, carbon dioxide is being reduced. Okay. So what do you mean by reduction over here? The addition of hydrogen atoms over here is called reduction. The removal of hydrogen atoms from the water molecule is called oxidation. Okay. So over here oxidized means the water will be oxidized to release oxygen. Okay. So this is what you have to remember. Removal of hydrogen and leads to the release of oxygen from the water molecule. So over here the carbon dioxide, the addition of hydrogen atoms over here, it will synthesize the carbohydrate from the carbon dioxide. This is what you have to remember. Whenever you are thinking about a carbon dioxide fixation, you have to remember one thing. This is a reaction. This is a continuous chain reaction of oxidation and reduction. So over here there are two major molecules involved, water molecule and carbon dioxide. Water is being oxidized, carbon dioxide is being reduced to the carbohydrate or sugar molecule. Okay. The question over here for you, what happens during the C3 plants or C3 cycle or the Calvin cycle? It has been discovered by Melvin Calvin. Okay. So as I told you, over here a carbon dioxide molecule is being fixed in a particular Calvin cycle. A carbon dioxide molecule enters into the C3 cycle or Calvin cycle. Okay. So the carbon dioxide molecule, it is being accepted by the ribulose bisphosphate. Okay. And it will enters into the particular cycle, and at one stage it will produce it will fix one carbon into a organic form. Okay, just remember one thing, the carbon dioxide as a gas is an inorganic form. Over here when it is combined with a particular hydrogen atom, it is going to be fixed as a organic molecule, as a sugar. But the point you have to remember over here, in each carbon cycle or each Calvin cycle, one carbon will be fixed in the form of organic form. Okay, this is what you have to remember. In each Calvin cycle, only one carbon will be fixed in the particular cycle. Okay, so the point you have to remember over here, as I told you, ribulose bisphosphate over here is a 5 carbon sugar. Okay, it will accept the particular carbon dioxide. Carbon dioxide has 1 carbon. Okay, 5 carbon plus 1 carbon, it will synthesize 6 carbon intermediate. Okay, this is a temporary molecule. This 6 carbon molecule immediately will form 2-3 carbon molecules. Okay, and it will enter into the formation of the organic compound. This is what you have to remember. The point you have to remember, where as I told you, In each cycle it will fix just one carbon at a time. Okay. The question over here, number of carbon cycles to generate one molecule of hexose. As I told you, hexose is a 6 carbon molecule. So you need 6 cycles to fix the particular hexose molecule or to form a particular glucose. One glucose, that is 6 carbon, you need 6 cycles of Calvin to perform. Okay, so you can easily eliminate 4, 9 and 8. Okay, this is how you have to answer the particular type of question from the Calvin cycle. Okay, so whenever you are thinking about a carbon dioxide fixation, in case of C3 plants, you have to remember one thing: each carbon dioxide will generate only one carbon fixed in the particular organic form. And see the next question. In C4 plants, carbon dioxide combines with, and see the choices: phosphoenol pyruvate, phosphoglyceric acid, phosphoglyceraldehyde and ribulose bisphosphate. So we have discussed the topic called carbon dioxide fixation. Okay, as I told you, the first discovered cycle in the carbon dioxide fixation is Calvin cycle. Who has discovered the particular Calvin cycle? As I told you, Melvin Calvin. Okay, and the point you have to remember over here, the Calvin cycle, we are calling the plants which perform the Calvin cycle are C3 plants. So why we are calling these as C3 plants? As I told you over here, the first forming product from the particular carbon dioxide fixation. So what happens over here in both the C3 and the C4 plants? What happens over here? The carbon dioxide fixation gonna happen. Okay, so they are gonna fix the atmospheric, the gaseous form of carbon dioxide into the organic form of sugar. Okay, this is what gonna happen in the C3 and C4 cycles or C3 and C4 plants. But the question over here, what is the difference between the C3 and C4? Okay, so in case of the carbon dioxide fixation, the carbon dioxide enters into the cycle and it will be fixed as a organic sugar molecule. Okay, so this is what you have to remember. The question over here, carbon dioxide combines with. Okay, so you have to remember the difference between the C3 and C4. Why we are calling the particular cycle as C3 and why we are calling that as a C4. Okay, so C3 means it refers to the first formed molecule from the particular cycle is 3 carbon molecule. This is what you have to remember. Okay, so in case of C3 cycle, the first formed molecule will be phosphoglyceric acid. Okay, so it will be a 3 carbon molecule. In case of C4, the first formed molecule will be 4 carbon molecule, that is oxaloacetic acid. In case of C4 plants, the first forming molecule over here is oxaloacetic acid. It is a 4 carbon molecule. Okay, so the question over here, what will be the acceptor molecule for the particular carbon dioxide in both C3 and C4? Okay, so you have to remember one thing. While we are discussing the previous question, as I told you, in case of C3 plants, the carbon dioxide will be accepted by ribulose bisphosphate. Okay, it is a 5 carbon molecule. It is going to accept the carbon dioxide. So it will produce a 6 carbon intermediate. From this, it is going to produce 3 carbon molecules. Okay, in case of C4 plants, first acceptor molecule for the particular carbon dioxide will be phosphoenol pyruvate. Okay, there will be no involvement of ribulose bisphosphate as a first acceptor, primary acceptor for the particular carbon dioxide molecule. Okay, so see the question over here, C4 plants carbon dioxide combines with, you can easily eliminate the ribulose bisphosphate. As I told you, ribulose bisphosphate is a primary acceptor of carbon dioxide in case of C3 plants. Okay, so your question is about C4 plants, so you can easily eliminate the ribulose bisphosphate. Just remember one point, ribulose bisphosphate is also one of the major enzyme involved in C4 plants also, but these are not the particular primary acceptor. Okay, they are involved in the particular C4 plants, but in a second phase of the particular cycle. And phosphoglyceraldehyde is not a correct answer. you must choose phosphoenol pyruvate and phosphoglyceric acid. As I told you, phosphoglyceric acid is a product formed from the C3 cycle, and the answer over here, phosphoenol pyruvate. Okay, this is how you have to choose the answer for this kind of question. And see the next question. In C4 plants, carbon dioxide fixation is done by. Okay, this is also related to the previous question. This question is talking about the location at which the particular carbon dioxide fixation gonna happen. Okay, and sclerenchyma cells, chlorenchyma cells, mesophils and guard cells. Okay, so we all know we are talking about the photosynthesis, and it should be the cells which are related to the particular photosynthetic system. Okay, the sclerenchyma, chlorenchyma, they are all related to the storage function of the particular plant, so you can easily eliminate the particular type of cells, and the answer must be in between mesophils and guard cells. Okay, so what are guard cells? As we all know, in case of the leaves, okay, they have some apertures or opening, okay, stomata opening, and every stomata will be surrounded by two cells, and those cells are called guard cells. Okay, so they are involved in the opening and closing of the particular stomatal apertures. But the question is about the carbon dioxide fixation. Okay, so the carbon dioxide fixation, the cells involved in the carbon dioxide fixation are the mesophil cells, not the guard cells. Okay, so you have to choose the answer mesophil cells. And what and all the process involved in the mesophil cells, we are going to discuss those later while we are discussing some other questions in the same topic. Now see the next question. The reaction center during the photosynthesis. Okay, so it is talking about the word reaction center. Carotene pigments, phytochrome pigments, P700 and cytochromes. Okay, and we will recall the particular reaction center. As I told you, the entire photosynthesis is going to start with the absorption of particular photons from the sunlight, that is light energy. Okay, so the photons will be observed over here by means of pigments involved in the particular photosystem 1 and 2, PS 1 and 2. Okay, so the point you have to remember over here, as I told you, as we have seen in the introduction classes, they are going to observe the particular light wavelength in between the 600 and 700. Okay, that is in a visible range. In the visible range, normally they are going to observe red and blue light regions. Okay, and this is what you have to remember. 600 to 700 nanometer range they are going to observe, and for each system, for PS1 and PS2, they have their specific reaction center. So reaction center, which means over here they have some specific or major pigment they are going to observe, or they are going to involved in the absorption of particular light energy. Okay, as I told you, it's made up of so many pigments. Chlorophyll A, B, xanthophylls, okay, there are so many pigments over here. But the point you have to remember over here, all the energy, all the photons is being absorbed by the particular entire pigment system will be transferred, or it will be relocated to the particular reaction center. There are two reaction centers are there, P700 and P680 for each PS1 and PS2. Okay, so this P700 or P60, they are going to act as a reaction center, and they are going to absorb the particular energy, and they are going to excite the particular molecules, and with this action only they are going to release the particular electrons for the further cycle in the photosynthesis. Okay, so the point you have to remember over here, the reaction center, it is always talking about the pigment system, but over here they have given you choices as pigments. Phytochrome is a pigment, carotene is a pigment, but you have to choose. Whenever the choice P700 or P680 is available, you have to eliminate the other pigments. Okay, so the reaction center, whenever you are talking about the reaction center, you have to think about P700 or the P680. Okay, so this is the answer for the particular type of question. Reaction center over here is P700. Okay, you may get the question specificity. What kind of the P center or reaction center in photosystem 1 or what kind of reaction center in photosystem 2. Okay. So in those type of question you have to answer whether it is P680 or P700. Okay. And see the next question. So over here a photosynthesizing plant is going to release O18 more than a normal oxygen. Okay. So this is your question and this is your statement. The question over here, the plant must have been supplied with. Okay. So it is talking about a process of the photosynthesis. Okay. So over here the particular plant, the particular photosynthesizing plant, it is going to release oxygen in a form of O18. Okay. So what is the normal form of oxygen? O16. Okay. So over here it is going to release oxygen isotope O18. Okay. The question over here, the plant must be supplied with what kind of molecule in a list over here and which molecule or which substitution going to produce the particular O18 as the product or evolution from the particular photosynthesis reaction. And see the choices O3. O3 is nothing but a ozone, and a second choice water with O18 isotope, carbon dioxide with O18 and sugar with O18. Okay. And you have to remember one major point in case of photosynthesis. What is photosynthesis? Here we are going to utilize carbon dioxide and water and they are going to produce sugar. This is what you have to remember. Whenever you are thinking about a photosynthesis, carbon dioxide will be utilized, water will be utilized to produce the particular sugar molecule. Okay. So reactants are carbon dioxide and water, products are sugar. Okay. So supplied with is talking about the reactants. So you can easily eliminate the sugar is being substituted with the particular oxygen because sugar is the product going to produce from the particular photosynthesis. You can easily eliminate. And ozone will not be the correct answer because there will be no the isotopic differences mentioned over here. So you can easily eliminate the ozone. The question over here is in between the water and carbon dioxide. What is the statement given for you? The oxygen is being released as O18. Okay. So the question must be like this. What is the source of the particular released oxygen from the particular photosynthesis? We all know during the photosynthesis the plant gonna release the oxygen or the oxygen will be evolved from the particular photosynthesis. The question over here, what is the source of the particular oxygen? Whether it is a carbon dioxide or it is a water molecule. This is the straight question from the particular topic. Okay. So both the carbon dioxide and water will contain the oxygen atom. Okay. So the question over here, what is the actual source of the particular oxygen for the particular photosynthesis reaction? Okay. So over here you have to remember one thing. Among these two choices, water with O18 or carbon dioxide with O18. Okay. So the key point you have to remember in the photosynthesis, the oxygen evolution from the photosynthesis is from the splitting of water. Okay. So as I told you, the light, they are going to release the photons, the excitation of photons, they are going to release the excitation of molecules, they are going to release the electrons. Okay. The water will be split into molecule, the atoms. Okay. H plus protons plus electrons plus oxygen. Okay. These are all the products from the water splitting. Okay. Splitting of water leads to protons plus electrons plus oxygen. As I told you, the protons and electrons will be utilized for the synthesis of ATP and NADPH. In the light dependent reaction, the oxygen will be released from the particular reaction, water splitting. Okay. So the answer over here is the water is being labeled with the particular oxygen isotope. Okay. So the carbon dioxide, what may be the use of carbon dioxide? As I told you, the carbon dioxide is going to be fixed or the carbon dioxide is going to be reduced. That means you're going to add hydrogen atoms so that you're going to synthesize carbohydrate from the carbon dioxide. Okay. So the oxygen atoms in the carbon dioxide gonna utilize for the formation of sugar molecule. Whenever you are observing the product, okay, suppose you are going to use the carbon dioxide is being labeled with oxygen 18, you will see the particular radio labeled oxygen in the sugar molecule, not in the evolution of the free oxygen from the particular reaction. Okay, so the answer over here is water with O18. Whenever you are using a water with O18, you will see the particular oxygen in a released gas form from the particular photosynthesis. Okay, and see the next question over here. The PS2, okay, photosystem 2 occurs in, okay, so this is talking about the site or location of the particular photosynthesis and photosystem 2. So as I told you, there are two types of reaction in the photosynthesis, light dependent and dark reactions. Okay, so both the reaction going to happen at a organelle called chloroplast. We all know that chloroplast is a photosynthetic organelle of any system. So over here, the chloroplast going to involved in the photosynthesis, and it is a organelle, that means a double membrane bound structure. So within a particular membrane bound structure, there is a semi-liquid matrix called stroma. Within a stroma, there are structures called lamella, and within a lamella, there are the coin-like arrangement called thylakoid, and the thylakoid going to form arrangement of grana. Okay, and you have to remember one thing. I told you light and dark reaction. All the dark reaction or the enzymes specific for the dark reactions are present in only in a stroma or liquid matrix of the particular chloroplast. Okay, all the light dependent reactions are present only in the membranes of the particular center organelles. Okay, so this is what you have to remember. Only the membranes are going to participate in the light dependent reaction. All the dark reactions going to participate or they are going to present in a particular stroma or semi-liquid matrix of the particular chloroplast. What will happen during the light reaction? As I told you, absorption of light, splitting of water, okay, and they are going to produce the ATP and NADPH during the light reaction. What will going to happen in a dark reaction? They are going to utilize the ATP and NADPH to fix the carbon dioxide as a organic sugar molecule. Okay, and see the choices over here: stroma, cytochrome, grana, and mitochondrial surface. You can easily eliminate the mitochondrial surface. This is completely different organelle from the photosynthesis. And see the choices, remaining choices: stroma, cytochrome, and grana. Cytochrome is a kind of molecule or complex involved in the electron transport chain. Okay, so you can easily eliminate this. The answer choice must be in between stroma and grana. I told you stroma is always involved in a dark reaction. There will be no relation between the PS2 and dark reaction. Also, you can easily eliminate the stroma. The answer choice must be the grana. Grana is nothing but the modification or the arrangement of the thylakoid one above the other. They are going to form the grana, and in the grana, the inside, the inner side of the membrane possesses the particular PS2 system. Okay, so the photosynthesis system, the photosystem 2 occurs in the grana or the membranes of the thylakoid. Inner side of the thylakoid membrane will have the particular PS2. And see the last question over here. The enzyme that catalyzes the carbon dioxide fixation in C4 plants. This is the clue for you. And choices: ribulose bisphosphate carboxylase, ribulose diphosphate, phosphoenol pyruvate carboxylase, and carboxy dismutase. Okay, and this is talking about an enzyme, so you can easily eliminate this. This is not an enzyme. So there are three remaining choices: ribulose bisphosphate carboxylase, phosphoenol pyruvate carboxylase, and carboxy dismutase. So while we are discussing the questions in a previous section, as I told you, there are two types of plants, C3 and C4. In case of C3 plants, okay, the receptor molecule or acceptor molecule for the carbon dioxide is ribulose bisphosphate. Over here it is phosphoenol pyruvate in case of C4 plants. So So this is a molecule going to accept the carbon dioxide. The enzyme which catalyzes the reaction over here is ribulose bisphosphate carboxylase. So other name for this is Rubisco. Okay, so why we are calling this is Rubisco? Ribulose bisphosphate carboxylase and oxygenase. That is why we are calling this as Rubisco. So over here, as I told you, the primary acceptor is not a ribulose bisphosphate. Over here is phosphoenol pyruvate. So the phosphoenol pyruvate, this reaction, the carbon dioxide fixation by the phosphoenol pyruvate will be catalyzed by the enzyme PEPCO. PEPCO refers to phosphoenol pyruvate carboxylase and oxygenase. Okay. So the question over here is the enzyme that catalyzes the carbon dioxide fixation in C4 plants. In case of C4 plants, you have to always remember one thing. The primary carbon dioxide acceptor over here is phosphoenol pyruvate. So answer must be, you have to easily eliminate the ribulose bisphosphate carboxylase. Carboxy dismutase you can easily eliminate. The phosphoenol pyruvate carboxylase or other term, name for this is PEPCO. Okay. So the PEPCO or the phosphoenol pyruvate carboxylate is the answer for the particular type of question. So in the next class we are going to continue the discussion of the same topic, the photosynthesis in plants.